Hi students, today we are going to learn about physiological anatomy, functions and lesions of the hypothalamus. Coming to the history, the central brain region of interest for neuroendocrinology is the hypothalamus. It is the name coined by Wilhelm His in 1893. Hypothalami, the name took from ancient Greek. Hypo means under, thalamus. Thalamus means chamber. Okay. It is in the size of an almond and it weighs 10 grams. Its function is one of the most important functions is to link the nervous system to the endocrine system via the pituitary gland. So it is the most important function of the hypothalamus to link the nervous system with the endocrine system. Hypothalamus is responsible for regulating certain metabolic processes and other activities of the autonomic nervous system. It also synthesizes and secretes certain neurohormones called releasing hormones or hypothalamic hormones. And these in turn stimulate or inhibit the secretion of the hormones from the pituitary gland. The hypothalamus controls body temperature, hunger, and important aspects of parenting and maternal attachment, behavior, thirst, fatigue, sleep, circadian rhythms, and is important in certain social behaviors such as sexual and aggressive behaviors. Coming to the boundaries of the hypothalamus. Superiorly, it has hypothalamic sulcus. Okay. Superiorly, it has hypothalamic sulcus. Inferiorly, it has floor of the third ventricle and tuber cinereum, infundibulum, okay, infundibulum, and the mammillary bodies, which are considered its parts only. Okay. Then medially, it has wall of third ventricle. Laterally, it has internal capsule. Anteriorly, it has anterior commissure and lamina terminalis. This is lamina terminalis. Posteriorly, it merges with the ventral thalamus. Okay, this is the thalamus. Posteriorly, it merges with the ventral thalamus. Coming to the embryological development of the hypothalamus. The hypothalamus develops from most anterior end of the developing neural tube. Most of it is derived from ventral part of the diencephalon, but its most rostral component, the preoptic area, develops from the telencephalon. So, most of it is derived from the diencephalon only. But the most rostral component, the preoptic area, is derived from the telencephalon. It is bounded anteriorly by the optic chiasm, laterally by the optic tracts, and posteriorly by the mammillary body. It is surrounded by the blood vessels of the circle of Willis. The hypothalamus is symmetrically duplicated on each side of the brain, with the third ventricle in the midline, forming a boundary between the two sides. For most functions, only the activity of one side of the hypothalamus is required. See here, this is prosencephalon or forebrain, which is again divided into telencephalon, from which cerebrum will be developed. This is diencephalon, from which thalamus, hypothalamus and epithalamus will be developed. This is one primary brain vesicle. There are three primary brain vesicles which will be developed into 5 secondary brain vesicles. So in a 3 to 4 week embryo, we have 3 primary brain vesicles, and in a 5-week embryo, we have 5 secondary brain vesicles. So prosencephalon is divided into telencephalon and diencephalon. From the telencephalon, cerebrum will be developed. From the diencephalon, thalamus, hypothalamus and epithalamus will be developed. From the mesencephalon, that is midbrain will be developed. From the rhombencephalon or hindbrain, it again divides into metencephalon, from which pons and cerebellum will be derived, and myelencephalon, from which medulla oblongata is formed. So there are some Key facts about the diencephalon. The parts are epithalamus, thalamus, subthalamus, metathalamus and hypothalamus. All these parts will be formed from the diencephalon. The blood supply is coming from posterior cerebral artery and posterior communicating artery. Okay, posterior cerebral and posterior communicating arteries. Embryology at 5th week of gestation, it is derived from prosencephalon. So here you can see at 5th week of gestation, it is derived from prosencephalon. The function is, it acts as a primary relay center and a processing center for sensory information and autonomic control. Okay, it has its connection with the limbic system. Limbic system is a seat of memory and emotion, and it has its connection with basal ganglia for motor coordination, and it has connections with primary sensory areas such as auditory and visual areas. Then coming to blood supply of the hypothalamus. The hypothalamus is supplied by the circle of Willis. Okay, this is the circle of Willis. The anteromedial branches of the anterior cerebral artery. Here they are showing it as A1. It is anteromedial branches of the anterior cerebral artery. That is A1. Then it is also supplied by posteromedial branches of the posterior communicating arteries. This is posterior cerebral artery. Here you have posterior communicating artery. So the posteromedial branches from the posterior communicating artery and the thalamo-perforating branches of the posterior cerebral artery, P1. This is posterior cerebral artery, P1. So, blood supply of the thalamus is from anterior cerebral artery, A1, that is anteromedial branches, and posterior communicating artery branches, posteromedial branches, and P1 from posterior cerebral artery, thalamo perforating branches. Okay, so from here A1 and from here posterior communicating artery and from here P1. Okay, here you have thalamus. Then physiological anatomy of the hypothalamus. From medial to lateral, it is divided into two zones, that is medial zone and lateral zone. From anterior to posterior, it is divided into four regions. They are preoptic region, supraoptic region, tuberal region and mammillary region. Preoptic, supraoptic, tuberal and mammillary region. Here you can see. This is the medial zone. This is the lateral zone. This is preoptic, supraoptic, tuberal and mammillary region. In between these two, we have third ventricle. So, some nuclei will be present in the hypothalamus. So, what are those nuclei? So, this is a schematic diagram to show the zones and regions of the hypothalamus and nuclei within them. So, you can see this table. It is divided into medial zone and lateral zone. Okay. The preoptic region is composed of preoptic nucleus. Okay. The preoptic region is composed of preoptic nucleus which is situated both in the medial zone and lateral zone. Then coming to supraoptic region. In the supraoptic region, in the medial zone, we have paraventricular nucleus, periventricular cell groups, suprachiasmatic nucleus and intermediate cell groups, or it may be called as anterior nucleus. So, in the supraoptic region, you have paraventricular nucleus and periventricular and suprachiasmatic nucleus and some intermediate cell groups. In the lateral zone, we have supraoptic nucleus. Okay, supraoptic region. In the lateral zone, we have supraoptic nucleus. Coming to the tuberal region, in the medial zone, we have dorsomedial nucleus, ventromedial nucleus, arcuate or infundibular nucleus, and premammillary nucleus. And in the lateral zone, we have lateral tuberal nucleus. Coming to the mammillary region or posterior region, we have medially posterior nucleus. Okay, posterior nucleus. And in the lateral zone, we have tuberomammillary nucleus. Okay, and mammillary body has mammillary nuclei. So here you can see this is the medial zone. This is the lateral zone. Okay, this is the preoptic nucleus, the preoptic region. And here you have supraoptic, means paraventricular nucleus, anterior nucleus, suprachiasmatic nucleus. Okay, suprachiasmatic nucleus. Okay, paraventricular, anterior and suprachiasmatic nucleus, and laterally you have supraoptic nucleus. Then coming to the tuberal region. Okay, this is supraoptic region, this is tuberal region. Tuberal region has medially dorsomedial nucleus, ventromedial or ventromedial nucleus. Okay, then you have infundibular nucleus. Okay, or arcuate nucleus and premammillary. This is premammillary nucleus. Okay, and laterally you have lateral tuberal nucleus. Okay, in the tuberal region, laterally you have lateral tuberal nucleus. Coming to mammillary region, we have posterior nucleus. Okay, and in the lateral zone, we have tuberomammillary nucleus, and this is mammillary body, which is having mammillary nuclei. Clear? Here you can see this is median preoptic nucleus. So preoptic nucleus is divided into two subregions, median and lateral. The median preoptic nucleus is important. It is helpful for generating the thirst. It responds to blood pressure changes which are caused by angiotensin II and regulates the thirst and fluid balance. Drinking water decreases the noradrenaline or norepinephrine release by the neurons of the median preoptic nucleus. Okay, so it is important for generating the thirst. The median preoptic nucleus is bounded laterally by the lateral preoptic nucleus and medially by the preoptic periventricular nucleus. It releases GnRH, that is gonadotropin releasing hormone, and controls copulation in males and is larger in males than in females, and is implicated in parental care in both males and females. Oxytocin and vasopressin are associated with maintaining the maternal care through local release in the median preoptic area and the adjacent bed nucleus of the stria terminalis, that is BNST. So oxytocin and vasopressin are associated with maintaining maternal care through the local release in the median preoptic area and BNST. Here you can see preoptic area divided into two subregions. You already know medial preoptic nucleus and area. So in all species which they are studied, it regulates the male sexual behavior. Lesions in this area can abolish the male sexual behavior. And sexually also it is dimorphic in size, that is larger in males. And it affects the onset of puberty. Okay. And hormones of pregnancy influence these neurons. Okay. Here for the onset of maternal behavior. And it synthesizes GnRH. And it is the site of oxytocin production and oxytocin receptors. And it is the site of AVP production and the gender specific levels of AVP receptors. So these are the functions of the medial preoptic nucleus. It controls the sexual behavior, it affects the onset of puberty, synthesizes GnRH, the size of synthesis of oxytocin, and AVP, that is arginine vasopressin. Coming to lateral preoptic nucleus or ventrolateral preoptic nucleus. This is ventrolateral preoptic nucleus. It mediates non-REM onset of sleep. REM means rapid eye movement. This is non-REM sleep. So it is important for going the person into non-REM sleep. Sleep is divided into REM sleep and non-REM sleep. So here the ventrolateral preoptic nucleus or intermediate nucleus, which is adjacent to the medial preoptic nucleus, it mediates the non-REM sleep. Okay? So sleep active neurons that fire least during wakefulness and most during non-REM sleep. Adenosine acts on these neurons and it will promote sleep and fatigue. Okay. The ventrolateral preoptic nucleus opposes the reticular arousal system, which promotes wakefulness. So, in our body, we have two systems to maintain wakefulness and to induce sleep. So, the reticular activating system, that is RAS, it will promote wakefulness, whereas VLPO, that is ventrolateral preoptic nucleus, it will promote non-REM sleep. Suprachiasmatic nucleus influences the VLPO directly through the thalamus to tie sleep pattern to circadian rhythm. Okay, suprachiasmatic lesion, if there is a lesion, it abolishes the connection. Okay, so you have understood that the ventrolateral preoptic nucleus VLPO is helpful for inducing non-REM sleep. Its action is opposite to the RAS system and it is through adenosine release in those neurons. And the suprachiasmatic nucleus is connected to VLPO. Why it is connected? To tie the sleep pattern to the circadian rhythm. So you can see preoptic periventricular nucleus. Okay, so you have subregions in the preoptic area, three subregions: preoptic periventricular nucleus, VLPO or ventrolateral preoptic nucleus, and medial preoptic nucleus. Okay. There will be rhythmic clock gene and clock controlled gene activity, where in the core and shell suprachiasmatic neurons and its astrocytes. It will release neurotransmitters and some diffusible molecules. Okay, and the output will go to mainly to hypothalamus, and it will also go to thalamus, ventral and midline, and the basal forebrain. Then coming to supraoptic and paraventricular nucleus. This is supraoptic and this is paraventricular nucleus. This is pituitary gland, anterior lobe and posterior lobe. The cell bodies of the neurosecretory cells are in the paraventricular and suprachiasmatic nuclei of the hypothalamus. So, the cell bodies are present in the nuclei of the hypothalamus, that is supraoptic and paraventricular nuclei. Their axons, they will descend down towards the posterior pituitary in the form of hypothalamo-hypophyseal tract. Okay. This tract begins in the hypothalamus and ends near the blood capillaries in the posterior pituitary. The paraventricular nucleus synthesizes the hormone oxytocin. Okay. And the supraoptic nucleus, it releases the hormone ADH, that is antidiuretic hormone, and it is also called as vasopressin. Okay. So here you can see these are the cell bodies and this is the tract, and it will end on the blood vessels on the posterior pituitary. So these hormones will enter into systemic circulation. Now coming to mammillary nuclei. They are helpful for recollective memory. Okay. The theta waves activates the CA3 neurons in the hippocampus via the fornix. Okay. Via the fornix, memory is transmitted to mammillary nuclei. Here you can see this is mammillary nuclei, medial mammillary nucleus and lateral mammillary nucleus. Okay. So here also you can see medial mammillary nucleus and lateral. This green one is the lateral mammillary nucleus. This blue one is the medial mammillary nucleus. Okay. These are the mammillary nuclei, two nuclei. So it is important for recollective memory. So from where it gets activated? From the theta waves. Okay, theta waves coming from the CA3 neurons which are present in the hippocampus. Which are present in the hippocampus. They will activate these things. Okay. How? Via the fornix. This memory is transmitted to mammillary nuclei. So you can see here the thalamus, here the pons, this ventral diencephalon. Okay. This is mammillothalamic tract. Okay. This is from mammillary region to premammillary nucleus. And this is from the dorsal thalamus to mammillary nuclei. From thalamus also it is having connection. And from the hippocampus also it is having connection. This is hippocampal formation. Subiculum, entorhinal cortex. Okay. From there information will go to the mammillary nucleus via the fornix. This is also hippocampal formation. Subiculum. From there, from the subiculum, the information will go to lateral mammillary nucleus and to the medial mammillary nucleus, and it also will get the information from the entorhinal cortex. Now coming to the connections of the hypothalamus. What are the afferent and efferent connections? Coming to the afferent connections, from the other parts of the limbic system, it will get the connection. That is medial forebrain bundle, fornix, stria terminalis, cingulate gyrus. The medial forebrain bundle is a neural pathway containing fibers from the basal olfactory regions, periamygdaloid region and the septal nuclei. Okay. And fibers from brainstem regions, that is ventral tegmental area and nigrostriatal pathway also. Okay. So what is medial forebrain bundle? It is a neural pathway and it has fibers from all these regions. What are those regions? They are from the basal olfactory regions, periamygdaloid region, septal nuclei and from brainstem regions, mainly ventral tegmental area and nigrostriatal area. You see, this is medial forebrain bundle. Here you can see this is medial forebrain bundle. Okay, and it is a part of the reward system and is involved in the integration of reward and pleasure. Electrical stimulation of the medial forebrain bundle is believed to cause sensations of pleasure. Okay, Microdialysis shows reinforcing electrical stimulation of the medial forebrain bundle using this method has shown to cause a release in the dopamine in the nucleus accumbens. Now coming to fornix. The fornix, what is fornix? This is the fornix. The fornix is a white matter tract that connects the hippocampus to several subcortical brain regions. Okay. So, it connects the hippocampus to several subcortical regions. And it is playing a pivotal role for episodic memory function. Functionally, the fornix transmits essential neurotransmitters as well as theta rhythms to the hippocampus. Okay. So this is fornix, fornix, the dotted one. Okay. From the hippocampus to several subcortical regions. It is helpful for episodic memory. Now coming to the stria terminalis or bed nucleus of the stria terminalis. This is stria terminalis or bed nucleus of the stria terminalis. This green one is a heterogeneous and complex limbic forebrain structure. It plays important role in controlling autonomic, neuroendocrine and behavioral responses. Coming to cingulate gyrus. The cingulate gyrus is an arch-shaped. This is an arch-shaped convolution which is situated just above the corpus callosum. The frontal portion is termed as the anterior cingulate gyrus, and it is a component of the limbic system and involved in processing the emotions and behavioral regulation. And it also helps to regulate the autonomic motor function. So, we are studying connections of the hypothalamus, afferent connections from other parts of the limbic system. In that we have studied median forebrain bundle, fornix, stria terminalis and cingulate gyrus. Now we are coming to the second connection, that is from the brain stem. So from the brain stem, mammillary peduncle, that is connecting tegmentum to mammillary body. Okay. So the tegmentum is connected to mammillary body through mammillary peduncle. It carries gustatory and general visceral afferents. Next one is dorsal longitudinal fasciculus of Sludge. Dorsal longitudinal fasciculus. It carries visceral afferents. Then medial forebrain bundle. It also carries visceral afferents. Then catecholaminergic pathways from locus ceruleus. It modifies the degree of alertness and regulates the output of releasing of hormones. Okay, catecholaminergic pathways from locus ceruleus and serotonergic pathways. These are helpful for sleep-wake cycle regulation. Okay, sleep-wake cycle regulation. So, these are the main afferent connections. So, from the brainstem, you have these 5 connections. Mammillary peduncle, dorsal longitudinal fasciculus of Sludge, medial forebrain bundle, and from the locus ceruleus, catecholaminergic pathways and serotonergic pathways. And it also gets afferent connections with the from the neocortex, globus pallidus, thalamus and retina. Then coming to efferent connections, it is sending its information to autonomic centers, limbic system, thalamus, tegmentum of the midbrain, neocortex and pituitary gland. Okay. Particularly hypothalamo-hypophyseal tract and tubero-infundibular tract. Coming to hypothalamo-hypophyseal tract. So, these two tracts, that is hypothalamo-hypophyseal tract and tubero-infundibular tract, they are connecting hypothalamus to the posterior pituitary. Okay. So, they are the efferent connections to the pituitary. So, this we have already discussed this. The hypothalamo-hypophyseal tract is a bundle of axons which connects the hypothalamic nucleus, that is supraoptic and paraventricular nucleus, to the posterior pituitary. Okay. So, the ADH, that is vasopressin or antidiuretic hormone, and oxytocin will be released into the circulation through this. Okay. Here they will be stored and released when they are needed. Coming to tubero-infundibular tract. So, this tubero-infundibular pathway, it is shown in the blue color. Okay. It also connects the hypothalamus with the pituitary gland. This is tubero-infundibular pathway. Okay. Tubero-infundibular pathway. This tubero-infundibular pathway refers to a population of dopamine neurons that projects from the arcuate nucleus, also called as infundibular nucleus, which is present in the tuberal region of the hypothalamus, the median eminence, to the median eminence. It is one of the four major dopamine pathways in the brain. So, we have four major dopamine pathways in the brain. Okay. It is one of this pathway, that is tubero-infundibular tract. So, this is started from the arcuate nucleus. It is also called infundibular nucleus. Okay, infundibular nucleus, which is present in the tuberal region of the hypothalamus. I hope you remember the tuberal. We have classified this into from anterior to posterior. The hypothalamus is classified into preoptic, supraoptic, tuberal and mammillary region. In the tuberal region of the hypothalamus, okay, we have in the medial zone, we have arcuate nucleus, also called as infundibular nucleus. From the cell bodies will be present in those nucleus, and the tract will be coming from those cell bodies. Okay, it is coming up to the median eminence. Okay, it is coming up to the median eminence. So, it will release dopamine. It is one of the four major dopamine pathways in the brain. Okay. Dopamine released at this site inhibits the secretion of prolactin from anterior pituitary gland lactotrophs. So, it is coming to anterior pituitary. This anterior pituitary is having many cell groups. In those, one group is lactotrophs. They will release prolactin hormone. But this dopamine will inhibit the secretion of prolactin. Okay. So, this is pituitary. Is coming like this. Okay. Then coming to the functions of the hypothalamus. What are the functions of the hypothalamus? First function is autonomic function. In that, it will regulate the cardiovascular system, that is posterior and lateral parts. They will if they are stimulated, they will increase the heart rate, blood pressure and cutaneous vasoconstriction. If preoptic nucleus is stimulated, it decreases the heart rate, blood pressure and it causes cutaneous vasodilatation. And it will regulate the pupil size. That is, stimulation of the posterior and lateral will cause dilation of the pupil, and the stimulation of the anterior and medial side of the nuclei will cause constriction of the pupil. And it will regulate the peristaltic and secretomotor function. This posterior and lateral nuclei will diminish the movements, whereas anterior and medial will increase the peristaltic and secretomotor movements. Then coming to endocrinal functions. It will control the anterior pituitary by tubero-infundibular tract, and it will control the posterior pituitary with the help of hypothalamo-hypophyseal tract, and it will regulate the uterine contractility and milk ejection from the breast, and it will regulate the sleep-wake cycle. That is, if anterior one is stimulated, it is causing sleep. If posterior one is stimulated, it wakes up the person. Then next function is control of circadian rhythm. It is by with the help of suprachiasmatic nuclei. It is also called biological clock. We have already seen. The inputs are coming from retina via retinohypothalamic fibers and from the lateral geniculate nuclei. This is a time-keeping system or biological clock, and it allows the organism to anticipate and prepare for the changes which are occurring in the physical environment, okay, that are associated with the day and night, thereby ensuring that the organism will do the right thing at the right time of the day. So here you can see this is a suprachiasmatic nucleus. From the retina, retinohypothalamic tract is coming. The output is going to cause affect the sleep-wake cycle. Okay. So from the retina, the impulses are coming from retinal ganglion cells. So upon light exposure on the retina, retinal ganglion cells will be stimulated, and it will send impulses via the retinohypothalamic tract. Okay. Then coming to fifth function, regulation of food intake. The lateral one will cause feeding, and ventromedial one will cause satiety. Okay. Via the glucostatic, lipostatic, thermostatic and gut peptide theories. Sixth one is the regulation of sexual behavior and reproduction. The tuberal region maintains the basal secretion of the GnRH hormone. Seventh function is the role in emotional and instinctual behavior. That is, lateral and ventromedial nuclei will affect the reward system, and if we stimulate the medial nuclei, it results in punishment. Here you can see, if a person is there, if the water is insufficient in the body, then there will be decrease in the blood volume and increase in the blood osmolality. So there will be dry mouth, and there will be stimulation of the osmoreceptors in the hypothalamus. So and there will be decrease in the blood pressure because of the decrease in the blood volume. The decrease in the blood pressure will increase the angiotensin 2 levels in the body. All these three, that is dry mouth, osmoreceptor stimulation in the hypothalamus, and increase in the angiotensin 2 levels in the body will stimulate the thirst center in the hypothalamus and increases the thirst. And the person will take water, and it decreases the blood osmolality. Okay. This is the thirst mechanism. And second one is the narcolepsy. It is hypothalamic disorder with abnormal sleep pattern. Okay. The patient will go to sudden attack of undesirable sleep during daytime in spite of having sufficient sleep in the night time. Okay. So this duration will be from few seconds to about 20 minutes also can be present. So it is a sleep disorder. Then coming to cataplexy. So cataplexy refers to sudden emotional burst of anger, fear or excitement, and it is associated with the narcolepsy. Okay. This attack also lasts for few minutes, but the conscious is not lost. Okay. So this cataplexy is sudden emotional outbursts of anger, fear or excitement. Sometimes it is connected with the narcolepsy. Then coming to Wernicke-Korsakoff psychosis syndrome. It is due to degeneration of the mammillary bodies because of the thiamine deficiency. Okay, it is mainly because of taking alcohol, and it has triad of ophthalmoplegia, ataxia, and global confusion. And this syndrome is an emergency condition in which 100 mg of thiamine given intramuscularly. Then the last lesion is confabulations. It is a memory disorder. So the person will tell fabricated, distorted words. Okay, and he will tell about the event which has never occurred. Okay, this is called confabulation. So lesions of the hypothalamus, there are 5 lesions: diabetes insipidus, narcolepsy, cataplexy, Wernicke-Korsakoff psychosis syndrome, confabulations. So here in the physiological anatomy, functions and lesions of the hypothalamus, we have learned about the history of the hypothalamus and the boundaries, embryological development, blood supply of the hypothalamus. Then we came to physiological anatomy of the hypothalamus, the classification of the hypothalamus from medial to lateral side and from anterior to posterior side. Okay? So that diagram we have to practice. Then we have seen about the nuclei in the medial zone and lateral zone of the hypothalamus and from the anterior to posterior region. Okay? So we have learned about each nuclei of the hypothalamus. So what is the function of each nuclei and where it is located? Then we have studied connections of the hypothalamus. What are the afferent connections and what are the efferent connections? Then we have studied the hypothalamo-hypophyseal tract and tubero-infundibular tract in detail, which is connecting the hypothalamus to the pituitary gland. Then we have learned the functions of the hypothalamus. They are nearly 10. Then we have seen the thirst mechanism flowchart. Then in the last, we have studied the lesions of the hypothalamus. The next video is about physiological anatomy and functions of the cerebral cortex.